Hello. All right, so today we're going to talk about the bacteria or the prokaryotes. And so we have covered some information that we need to know about evolution. And so the next step when we talk about the basically the living world is to start out with the simplest of the organisms. And so that's going to take us to the bacteria. As we've talked about evolution, we've got to think about those very first steps. What had to happen in that kind of um, world that was millions of years ago, and how did things start to come together, and how did life kind of start to happen? We don't know for sure, but we do have some hypotheses, some educated guesses on things that may have happened in order for these living organisms to kind of get started. And so one of the hypotheses is called the prebiotic soup hypothesis, and the other one is the iron sulfur world hypothesis. It's probably a combination of both because both of these things could have happened. The conditions were right for both of these things when you look back at the Earth millions of years ago. So we do know that early on in the Earth's history there was a very thin atmosphere with no oxygen, and so oxygen is obviously what's vital for us to be alive. So there had to be something that eventually, put oxygen in the air, but we weren't there yet. And so this early atmosphere had no oxygen, and it was what we call a reducing environment. So if you think about chemistry, that's just a chemical term. And there were some scientists who tried to do this in a lab where they basically tried to replicate the early atmosphere. And what they found is in this early atmosphere, because all the elements, that are in the Earth today were there then, and they were able to come together and join up and with bonding and make things like amino acids and other organic molecules. And so when I say organic molecule, that's something that has a carbon in it. And so we're a carbon-based life form. So you started to see just a spontaneous combination of elements forming these organic molecules like amino acids. And that happened just, think about the oceans and the watery environment. It was a building, there was volcanoes, it was kind of a hot, there was volcanic vents and hot spots on the Earth at that time. And so if you replicate that in a lab, you can also see that you can get the same production of those amino acids, and those amino acids will link together and form small proteins. So probably a combination of both of these things is how we got those first formations of these early organic molecules that are really the building blocks for life. So in order to get biologic life, the first, and to get biologic evolution, where you get the formation of simple organisms all the way up to more complex organisms, you have to have a chemical evolution first. And so the first stages in the steps toward life on Earth was to form these organic molecules from inorganic compounds. Okay, so again, elemental elements, hydrogen and carbon and nitrogen and iron and things like that had to come together and form organic molecules like amino acids. And then once you have an amino acid, they can join together to form these peptides and these proteins. So that's kind of the first step. The chemistry's got to be there first, because literally we are just a big bag of chemistry. So the first steps was to get the chemistry right. The next step was for these organic molecules to start to be surrounded and bound and individualized from other things. So we know that the basic unit of life is the cell. So we had to get something where we started to form a protocell, something that was an individual. And then came the next step where you actually have what we call the genetic blueprint, the presence of DNA and RNA. All right, so if you want to think about that in a visual way, look back to the early beginnings of the Earth, the sulfur and the heat from the volcanoes. You're going to have these inorganic molecules. They're going to be using the energy of the sulfur vents, the volcanic vents, the heat of, a lot of times you need heat for a reaction to occur. That heat is going to generate bigger molecules. You get the formation of these small organic molecules. Small molecules polymerize or join together to form big macromolecules. These macromolecules can spontaneously form cells. using a plasma membrane. So if you think about the phospholipid bilayer that you know about that makes up cells, the cell membrane, that can spontaneously generate. If you have a beaker and you put in phospholipids, they, due to their charges, they will automatically form basically a cell membrane. So they'll just do that just based on their chemistry and their charges. So basically you have a balloon or a bubble, a cell, and then eventually the presence of genetic material. So, this is where we are now with our hypothesis on how this early life came into being. So what we wind up with are the very first cells, okay? And these are going to be our prokaryotes, or our bacteria. And so that's what we're going to focus on our lecture today, are the prokaryotes. So this is a large group of organisms, and it's made up of, when you think about the bacteria, it's really two separate domains. Okay, so if you think about the whole tree of life, we have three domains. You have your bacteria, your archaea, and your eukarya. Okay, and this is just a stylized schematic tree, so you can see all of these are bacteria, all of these are archaea, and all of these are eukaryotes. So these two are what we would kind of call the bacterial world. The true bacteria, okay, these are your prokaryotes. These are the smallest ones, the most ancient. They don't have a nucleus. There's no membrane-bound nucleus. That's what prokaryote means. The archaea are kind of like specialized bacteria. They're your extremophiles. So they're just like bacteria as far as having no nucleus, being prokaryotes, but they live in very extreme environments. And so they can live in such extreme environments that we consider them two separate domains of organisms. Then the last domain, we've talked about this before, is the eukarya, and this is the large cells, large complex multicellular organisms. These are your plants, your animals, your fungi, everything that we know of as being living organisms that's not a bacteria. And they have organelles and a membrane-bound nucleus. So today we're focusing on these two. There are entire courses just on these organisms, but we're doing it in about 20 minutes. So this is just what you need to know about the bacteria and the archaea, or the prokaryotes. So prokaryotes are important. Number one, they're very quick to grow and develop and reproduce, okay, and mutate. That's one of the problems that we have with antibiotic resistance of bacteria. They can grow and reproduce and mutate so quickly and change so quickly. And what did we say about evolution? Evolution means change over time. They can do this very, very rapidly, and they can do it in response to very adverse conditions and in very adverse conditions. They can live in a lot of different places. We actually have some fossils of bacteria that are 3.5 billion years old. They can live with both oxygen and no oxygen, so aerobic, which means they require oxygen to live, and anaerobic, which means oxygen is deadly to them. So you think way back to those early times on the Earth's geologic time frame when we were just getting oxygen, so there would be areas that would be basically oxygen rich or oxygen poor. Bacteria were able to live in both of those. Same goes today. There are places on Earth and parts of our body and certain environments that don't have oxygen, and bacteria thrive there. The anaerobes, a lot of times they're the ones that can make you really sick. One of those would be like how you get botulism. It's an anaerobe that can live in a low oxygen or no oxygen environment. So they can be both anaerobes or aerobes. They can be autotrophs, which means they photosynthesize, okay, or they can obtain energy from the sun. So they can also be heterotrophs. I lost my train of thought there, which means they have to take food in and break it down for energy. So autotrophs make their own energy. Heterotrophs have to absorb and take energy in. Okay. So some examples of that would be like the things that basically are decomposers, things that feed on decaying material, things like parasites, things like that would be your heterotrophs. All right, so the domain bacteria, again, they're prokaryotes. They're one-celled, so that's what unicelled means. They have a cell wall that contains peptidoglycan, so that means they have a cell wall. Remember, one of the things that's different between like animals and plants, as plants have cell walls and animals don't. Well, bacteria have a cell wall that gives it strength. They have a certain type of membrane lipid, a little different than the kind of membrane lipids that we have. It's not branched, but it still is able to form a membrane around the organism. Examples of bacteria, there's all kinds. They're the ones that live on our body. They're the ones that we use to recycle waste, the ones that make us sick. And blue-green algae are actually a type of bacteria. So are they good, bad, or indifferent? Well, they can be both. Some are good, some are bad. There are a lot of bacteria that cause diseases and can be, even fatal to us. For example, Neisseria meningitidis causes meningitis, which is an inflammation of the membrane surrounding your brain and spinal cord. You don't want that. So it's a disease-causing bacteria that we try to avoid. But there are good bacteria, those that even make antibiotics for us. There's a bacteria called Actinomycetes that makes streptomycin, which we use as an antibiotic. So bacteria make things that kill bacteria. And sometimes we need them. There are bacteria that actually live in the guts of animals to help them out. Cellulose is something that we don't digest, but bacteria do. So when you eat plants high in cellulose, you're going to digest as much as you can, but what remains in your gut from that, gut bacteria can then break that down for you. That's one of the reasons, that's where gas comes from when you expel gas after eating food. E. coli is one of those bacteria that we actually do need because it helps us make vitamin K. So when it's doing its job inside of our gut, it makes vitamin K, and we have to have vitamin K in order to clot our own blood. So we must have that. So it's a symbiotic or helpful relationship that we have with bacteria. And you're literally covered with bacteria. There's all kind of normal flora and bacteria that you have on your body that is not causing you any harm. Okay? Do they do anything else besides thinking about health benefits? Well, they actually help us with our nutrition and with allowing plants to grow. We see that bacteria can live on the roots of some plants, and what they do is they convert nitrogen to something that's usable. So things like peanuts and soybeans, they have these little nodes that contain bacteria that actually help to put nitrogen into the soil. And when we talk about plants, you'll find out that nitrogen is like fertilizer. It's a key element that plants need to grow. And so you want to, when you think about crop rotation, when you see them rotating out corn and cotton and soybeans, the reason they do that is the soybeans put nitrogen back in the soil, and cotton and corn will need that. So the bacteria help us put the nitrogen back into the soil. It's called nitrogen fixation, but it's basically putting nitrogen in the soil so that plants can then absorb it through their roots and use it to grow. If you like yogurt, there's bacteria. Streptococcus thermophilus is one of those. There's several, but the good yogurt bacteria. A lot of times if you are sick, sometimes maybe with a digestive problem, you can eat eat yogurt, and those bacteria will kind of help get you back feeling good again. Again, our planet would be covered with dead stuff if we didn't have things to break that dead stuff down. And so the bacteria can help us break down organic matter, the saprobes. And we can also see them as the very beginning of the food web. So if you think about, the big fish eat the medium-sized fish, which eats the little fish, well, eventually it goes all the way down to the bacterial level. Okay, so the bacteria is kind of the very base of our food pyramid when you think about food webs and food pyramids for animals and organisms and plants. An example of bacteria are also the cyanobacteria. They're true bacteria. It's just a group of bacteria that are photosynthetic. Okay, so a lot of times you'll see these. If you see that green slime on a pond, that's you may think it's moss or algae or something, but it's actually cyanobacteria that form these long chains, and they can photosynthesize. And so they were some of the first organisms on Earth that could photosynthesize, and they produce a lot of oxygen. So they are thought to be the major contributors of making oxygen and kind of filling our atmosphere up with oxygen. Okay, so our oceans are important, water's important by having cyanobacteria, which make oxygen. So bacteria, obviously, if you have one bacteria, you're going to have two. They have to reproduce, and they can do it in one of several ways. They can either have sexual or asexual reproduction. So if you have sexual reproduction, we also call that conjugation, which is they basically send out a little probe and they directly transfer genetic information. So I just basically take my DNA and insert it into a death one. So they're transferring and sharing genetic information. Or asexually, there's something called binary fission, which means they just literally divide in half. And then so 1 becomes 2, becomes 4, becomes 8, becomes 16. All right? So 2 ways that bacteria can reproduce. They're also very tough. Okay, so bacteria, you have to think about this when you're thinking about planning for, like, especially disease processes and how you're going to beat the bacteria, because they're very resilient. And one of the things they can do is survive a long time by putting themselves into an endospore. Okay, so they can basically form something called an endospore, which is a kind of a large, kind of a hard shell basically around the bacteria. So here's the bacteria right here, and it's formed this kind of this coating on the outside like a shell. And so then they can survive this way, kind of in suspended animation, almost like hibernation, until conditions are more favorable, and then they kind of hatch out and they can become bacteria doing their thing again. So anthrax is one of those, Bacillus anthracis. So anthrax is notorious for forming these spores and can be there for hundreds of years. And then, like, think about permafrost, somewhere where it's really, really cold, the ground's frozen. The anthrax spores are just sitting there dormant. When it warms up, they wake up, they hatch, and you have anthrax. And so actually, as we've seen these periods of global warming that are going on right now, there are areas in the far reaches in the north where we have what's called permafrost, which means the ground never really thaws out, but it's thawing out now. And as it's thawing out, some of these spores are starting to wake up and hatch. And so we're seeing anthrax in places that we haven't seen before, because it was there at one time when the earth was warmer, and then when the conditions became too cold for it to survive, they kind of went dormant, and now as it's warming up, they're coming back. So we're seeing anthrax in some places that we didn't expect to see it, or haven't seen it in a while. This is just kind of I want to talk to you a little bit about shapes. When you look at a bacterial name, a lot of times it'll have like coccus or bacillus or spirillum as part of the name, or it'll be described that way, and that's describing the shape of it. So, like, I have a microbiology degree, so I think of all the cocci and I think of all the bacillus, and so that's just basically dividing the bacteria into categories based on their shape. So if it's a cocci or coccus, it's going to be round. If it's a bacillus, it's going to be kind of an oblong tic-tac shape. If it's a spirillum, it's going to be spiral shaped. All right, so if you ever see a name that has the cocci or bacillus or spirillum in it, that kind of tells you what it looks like. Also, if it's the coccus type bacteria, they can kind of join together like beads on a string, and a lot of times you'll find bacteria that come as two together. That's a diplococcus, or a long line, that's a streptococcus, or kind of in a bunch like a grape, that's a staphylococcus. So if you have a streptococcus infection, you've just got an infection of these long chains of these cocci bacteria, the staphylococcus infection, then you've got the type of bacteria that kind of form these bunches of grapes, kind of a structure. So these names actually tell you something. So next time you get a strep test is short for streptococcus. And so that means your bacteria are in a formation like that, a long chain of bacteria. Another thing you do if you wind up doing the health field and going into microbiology is you'll also have kind of a way to identify bacteria by staining it. And you can take your bacteria, take your sample, and you put this stain on it called a gram stain, and it kind of divides the bacterial world into plus or minus. There's gram positive, which means they stain purple, and there's gram negative, which means they are pink. And that stain works because of what the cell wall contains. So if it's gram-positive, it's going to have a peptidoglycan, a thick peptidoglycan layer. And if it's gram-negative, it's going to have this thin peptidoglycan layer. And no teichoic acid, where you do have that acid in the gram-positives. For my purposes, I just want you to know that we divide the world, the bacterial domain, into gram-positive and gram-negative. So if you do take a micro class, one of the first things you'll learn how to do is gram stain. And then you'll look at the bacteria and go, okay, are they purple? Are they pink? And the next thing you'll look at is, what shape are they? Are they gram-positive cocci? Are they gram-positive? People call these rods too instead of bacillus. Are they round? Are they rod-shaped? Are they spiral? So these are just some identification markers for bacteria. But you may see these terms every once in a while, like in a particular prescription. It may say works for gram-positive or does not work for gram-positive bacteria. And that'll tell you a little bit more about the drug you're taking and how effective it is for your particular disease. Penicillin is another antibiotic. It doesn't come from a bacteria. It actually comes from a mold. And so you can see here's a plate where we've streaked out some bacteria. Here's a little area of penicillin, and you can see it is killing all the bacteria around it. And this was kind of found by accident. Penicillin is, there's penicillin spores kind of all around. And so one day they left the plate, the bacteria plate was open, some spores landed on the bacteria on the plate, and then a couple days later they came back and realized whatever this thing was making was killing the bacteria. And they're like, oh, maybe we can use that as some way to treat infection. And that's basically the way that happened. Kind of a happy accident. So that gets us through one domain. So I told you we're doing an entire domain really fast. The second domain of the archaea, going to do it even quicker, just kind of tell you a little bit about it. Again, prokaryote, unicelled, but these live in extreme conditions like sea vents and hot springs, cold like in glaciers, and even out in the desert. So you may think there's no life in these places, but there's going to be some bacteria there and some of the harshest places on Earth. Which is why when we're looking at life on other planets like Mars, what we're looking for are bacteria, because as harsh as Mars is, we've got some areas here that are just as harsh. So if it can make it here, it can possibly make it on Mars as well. One of the difference between archaea and the bacteria is they do not have a peptidoglycan. They have a branch lipid in their cell membrane, and they have a unique RNA that's kind of unique, allows them a little special consideration compared to the other bacteria. So those are kind of your differences. So one group is the methanogens. They're anaerobic, which means they don't need oxygen. It's toxic to them. They make methane, or natural gas, as a waste product. So if you've ever been to a swamp and it's kind of bubbling up and the gases are being released, those are going to be those methanogens making gas from the swamp. You see them in sewage and landfills. We're now learning that you can put pipes into landfills and catch that gas and use it to burn and make energy. So you're getting the natural gas from landfills and trying to reharness the gas that's being made from decomposed garbage and use that for energy. So also it's a byproduct of sewage as well. So trying to come up by using these bacteria to our advantage to come up with renewable sources of energy from our waste. You have halophiles, and halo is basically the short term for salt. Okay, so these are aerobes, and they like salt, so you're going to find them in salty environments. Things like the Great Salt Lake of Utah, salt ponds. There's some around San Francisco Bay. So anywhere that's really salty, the salty environment, you'll see halophiles. They have a certain pigment in them. So when you do photosynthesis, you know, think about chlorophyll. It's a green pigment, and that's how you make sugars. They have a similar pigment, but it's called rhodopsin. And so when they do photosynthesis, instead of things being green, it's pink. But their type of photosynthesis does not make oxygen. So it's kind of a different pigment that they're based on. So when they go through their process of photosynthesis, it doesn't produce oxygen. The cool thing about this, because when we talk about evolution and we find things that work and they tend to be, carried on and move forward, and we can find similar proteins and enzymes, all throughout biologic life. Rhodopsin that's found in these extremophiles is also found in us. So our retina uses the same pigment, rhodopsin, as a way for us to see color, to see black and white, kind of our night vision. So this pigment is what we use in order for us to have night vision. And so if you take anatomy, we'll talk a lot about that. If you want to ask me questions about it in class, I'm happy to talk to you. But so rhodopsin's not just for extremophiles. We use it so we can see at night. So this is just a picture of one of these salt areas, and you can see this nice pinks and blues and purples from them using that rhodopsin instead of chlorophyll for photosynthesis. Okay, so it makes their stuff look purple instead of green. And the last group are your thermophiles. So temperature, they grow in hot places. They're also anaerobes. They do anaerobic respiration. They live in hot springs, anywhere where it's really, really hot. They can even live above boiling, the boiling point of water. That's pretty hot. They have value to us, not just because they have their life on Earth, but they also have some genes that allow them to be stable in heat. And so there's one called Thermus aquaticus, so hot water basically, that we find in the Yellowstone hot springs, and it makes something called Taq polymerase. And that TAQ is basically from the first letters of the name of the bacteria, that allows you to make copies of DNA. And so as a researcher, if you're in the lab and you need to make a lot of copies of DNA, you actually use this thing. So we actually are using something that was made in nature to help us in the laboratory to make copies of DNA for biologic research. So that's just pretty cool. So this is just an example of some hot springs in Yellowstone, and the color is based on the heat. The closer you get to the middle of the vent, the more blue it is, the darker blue that you see. So this is going to be hot, and then as you move out, it's a little bit cooler. And those colors are not as blue. Now, if you ever go to Yellowstone, unfortunately, a lot of the thermal vents don't look like rainbows anymore, because what has happened is people have started throwing stuff into the vents, like pennies to wish good luck or whatever, and so they block in the vents, and so now they're cooling off, and so now you don't have these gradations of temperature anymore. So some of our hot springs have been irreparably damaged because people are stupid for blowing or throwing things down into the hot vents as a wishing well. Which really irritates me when I see people throwing things in the hot springs. All right, and that's it. So that's all you got to know about bacteria. So I have done an entire degree. I'd have a 4-year degree in microbiology, and I did it in 28 minutes. All right, so thanks a lot, and I will see you in the next one.